Welcome to this scientific lecture on encapsulation in microfluidic devices. Today we're going to explore how we can trap materials inside tiny droplets or capsules using the principles of fluid dynamics at the microscale. Let me start by explaining what we mean by microfluidic encapsulation. This is the process of wrapping or surrounding a material, whether it's a cell, a drug, or a chemical reagent, inside a protective shell or droplet at the microscale level. Encapsulation refers to the act of trapping materials within protective barriers, typically ranging from 1 micrometer to several hundred micrometers in diameter. But why do we focus on the microscale? The reason is that at these small dimensions, the physics of fluids changes dramatically. Surface tension becomes dominant over gravity, and viscous forces dominate over inertial forces. Here we see a simplified diagram of an encapsulated system. The outer blue shell represents the protective barrier, while the inner region contains the material we want to protect, in this case cells shown as red dots. Let me ask you a question. Why does fluid behavior change at the microscale? The answer lies in a dimensionless number called the Reynolds number. The Reynolds number is defined as the ratio of inertial forces to viscous forces. It equals density times velocity times characteristic length divided by dynamic viscosity. At the macroscale, like in a river or a pipe, the Reynolds number is typically high, meaning inertial forces dominate and you get turbulent, chaotic flow. But at the microscale, the Reynolds number drops dramatically, often below 1. Viscous forces dominate and flow becomes completely laminar and predictable. This is crucial for controlled encapsulation. In microfluidic devices, Reynolds numbers are typically much less than 1, ensuring laminar flow conditions. Now let's discuss the main types of encapsulation we can achieve in microfluidic devices. There are 3 primary categories. First, we have droplet-based encapsulation. This involves creating water in oil droplets, or oil in water droplets, using immiscible fluids. Here we see water droplets flowing through an oil continuous phase in a microfluidic channel. Each droplet acts as an isolated microreactor. Second, we have hydrogel-based encapsulation. Here, cells or materials are trapped inside a polymer gel matrix that provides structural support. Third, we can create liposomes and vesicles, which are lipid bilayer structures that mimic cell membranes, and can encapsulate both hydrophilic and hydrophobic materials. Let me now explain the key physical properties that govern the encapsulation process. Understanding these is critical for designing effective microfluidic systems. The first property is interfacial tension. This is the force per unit length acting at the interface between 2 immiscible fluids. Interfacial tension gamma equals force divided by length. It's typically measured in millinewtons per meter. High interfacial tension is what makes droplets want to minimize their surface area, leading to stable, spherical shapes. This is why oil and water droplets are round. The second important property is the capillary number, which compares viscous forces to interfacial tension forces. The capillary number equals dynamic viscosity times velocity divided by interfacial tension. When the capillary number is low, surface tension dominates, and you get nice, stable, uniform droplets. This is the regime we typically operate in for encapsulation. When the capillary number is high, viscous forces dominate, and droplets can deform or break apart, which we generally want to avoid. The third crucial property is the contact angle, which describes how a liquid interacts with a solid surface. This diagram shows the contact angle theta, which is the angle formed where the liquid droplet meets the solid surface. When the contact angle is less than 90 degrees, the surface is hydrophilic, meaning water-loving. The droplet spreads on the surface. When the contact angle is greater than 90 degrees, the surface is hydrophobic, meaning water-repelling. The droplet beads up. By controlling the surface chemistry of our microfluidic channels, we can precisely control where and how droplets form. Now let me describe the common microfluidic geometries used for encapsulation. The geometry of the device determines the droplet generation mechanism. The first and simplest geometry is the T-junction. Here, the dispersed phase meets the continuous phase at a perpendicular intersection. In a T-junction, the blue dispersed phase flows downward and meets the orange continuous phase flowing horizontally. Droplets pinch off at the junction. The second geometry is flow focusing. This uses three inlets where the continuous phase flows on both sides of the dispersed phase, focusing it into a narrow stream. In flow focusing, two streams of oil pinch the water stream from both sides, creating very uniform droplets at high frequency. The third geometry is co-flow, where one fluid flows concentrically inside another fluid in the same direction. Among these geometries, flow focusing typically produces the most monodispersed droplets, meaning droplets with very uniform size distribution. Let me pose a question. What factors control the size of the droplets we produce? There are three main parameters. First, the flow rate ratio between the dispersed and continuous phases. Higher continuous phase flow rate produces smaller droplets. We define phi as the ratio of dispersed flow rate Q sub D to continuous flow rate Q sub C. Second, the viscosity ratio between the two fluids affects droplet formation dynamics. Higher viscosity in the dispersed phase generally produces larger droplets. Third, the channel geometry and dimensions directly influence droplet size. Smaller channels produce smaller droplets. Now let's discuss an absolutely critical component of droplet encapsulation: surfactants. These molecules are essential for stabilizing droplets. Surfactants are amphiphilic molecules, meaning they have both a hydrophobic tail that likes oil and a hydrophilic head that likes water. This diagram shows a surfactant molecule with its red hydrophilic head and blue hydrophobic tail. When many surfactants arrange at an interface, they form a protective layer around droplets. Surfactants work by absorbing at the oil-water interface and dramatically reducing the interfacial tension, making it easier to form droplets. More importantly, surfactants prevent droplet coalescence. Without surfactants, droplets would merge together, and we would lose our encapsulation. Common surfactants used in microfluidics include SPAN 80 for water and oil systems, TWEEN 20 for oil and water systems, and specialized fluorosurfactants for fluorinated oil systems. Let me now explain the chemistry behind hydrogel encapsulation, which is particularly important for biological applications. Hydrogels are three-dimensional networks of cross-linked polymer chains that can absorb and retain large amounts of water while maintaining their structure. Common materials include alginate, which is derived from seaweed, agarose from agar, and synthetic polyethylene glycol, known as PEE. Alginate gelation is triggered by divalent cations like calcium. When alginate chains encounter calcium ions, they cross-link and form a solid gel network. This diagram shows the hydrogel network structure, with green polymer chains connected by yellow cross-linkers, with red cells trapped inside the network. For microfluidic encapsulation, gelation must occur rapidly, typically within seconds, so that the gel solidifies before the droplet exits the device. Now let's explore the exciting applications of microfluidic encapsulation. This technology has revolutionized multiple fields. Application 1 is single-cell analysis. By encapsulating individual cells in droplets, we can study the heterogeneity of cell populations at unprecedented resolution. Each droplet acts as an isolated microreactor containing just one cell, allowing us to measure the specific genetic or protein expression of that individual cell without contamination from neighbors. Application 2 is drug delivery. Encapsulation protects drugs from degradation and enables controlled, sustained release at target sites in the body. Here we see a drug-loaded capsule with red drug molecules inside. Over time, the capsule breaks down and releases the drug in a controlled manner. Application 3 is enzyme immobilization. Encapsulating enzymes in droplets or gels protects them from denaturation and allows reuse in industrial processes. Application 4 is cell therapy and tissue engineering. Encapsulated cells can be implanted into patients to deliver therapeutic proteins or regenerate damaged tissue. Let me ask another important question. Why do we care so much about producing monodispersed droplets? Why does uniform size matter? First, uniform droplet size ensures consistent drug dosing. If droplets vary in size, patients receive unpredictable amounts of medication. Second, in research applications, uniform size is essential for reproducible experiments. Every droplet should be an identical microreactor. We quantify size uniformity using the coefficient of variation, which is the standard deviation divided by the mean diameter, typically expressed as a percentage. Coefficient of variation equals standard deviation sigma divided by mean diameter mu times 100%. Good microfluidic systems routinely achieve coefficient of variation values less than 5%, meaning highly uniform droplets. Let me introduce an advanced topic: double emulsions. These are droplets within droplets, creating core shell structures. This shows a water and oil and water double emulsion structure. The green inner core is water, surrounded by an orange oil shell, all suspended in a blue outer water phase. Double emulsions enable compartmentalized reactions where you can have incompatible reagents separated by the oil barrier. These structures are created using sequential flow-focusing geometries, where you form the inner droplet first, then encapsulate that entire droplet in a second step. Now let's discuss how we characterize and measure these encapsulated systems. Several techniques are essential. First, optical microscopy with high-speed cameras allows us to visualize droplet formation in real time and measure droplet diameters with micrometer precision. Second, flow cytometry can analyze thousands of droplets per second, measuring their size, fluorescence, and other optical properties. Third, fluorescence microscopy lets us visualize encapsulated materials like cells or fluorescent drugs inside capsules. Fourth, for hydrogel capsules, mechanical testing using microindentation or rheology reveals the stiffness and mechanical properties of the gel matrix. Despite the powerful capabilities of microfluidic encapsulation, several challenges and limitations remain. Challenge 1 is scaling up production. Microfluidic devices excel at producing uniform droplets, but throughput is limited. Producing clinically relevant quantities requires parallelization of many devices. Challenge 2 is long-term stability. Maintaining encapsulation integrity over weeks or months, especially for biological applications, remains difficult. Challenge 3 is biocompatibility. Many surfactants and materials used in microfluidics are toxic to cells, limiting biomedical applications. Challenge 4 is cost. Microfluidic devices, especially those made with soft lithography using cleanroom facilities, are expensive to produce and require specialized expertise. Looking toward the future, several exciting directions are emerging in microfluidic encapsulation research. Direction 1 is creating artificial cells and protocells. Researchers are using encapsulation to build synthetic cells from the bottom up, containing DNA, RNA, proteins, and metabolic pathways. Direction 2 is smart responsive capsules that can sense their environment and release cargo on demand in response to specific triggers like pH, temperature, or light. Direction 3 is integration with three-dimensional bioprinting. Encapsulated cells can serve as bioink components for printing complex tissue structures. Direction 4 is point-of-care diagnostics. Portable microfluidic devices that encapsulate and analyze patient samples could enable rapid disease diagnosis at the bedside. Let me work through a quantitative example to illustrate the physics. Suppose we have a T-junction device with a channel width of 100 micrometers. Let's define our system parameters. Continuous phase flow rate is 100 microliters per hour. Dispersed phase flow rate is 10 microliters per hour. Q sub C equals 100 microliters per hour. Q sub D equals 10 microliters per hour. The flow rate ratio phi equals 10 divided by 100, which equals 0.1. For T-junction geometry, droplet volume is approximately proportional to the flow rate ratio times a geometric factor related to channel dimensions. Droplet volume 5 sub D is approximately phi times W cubed, where W is the channel width. Plugging in our values, droplet volume is approximately 0.1 times 100 micrometers cubed, which equals 10 to the 5th cubic micrometers. Converting to diameter assuming a sphere, we use the formula D equals 2 times the cube root of 3V sub D over 4 pi. This gives approximately 58 micrometers. This calculation is approximate, but demonstrates how channel dimensions and flow rates determine droplet size in a predictable manner. Let me give you an analogy to make this concept more intuitive. Think of microfluidic encapsulation, like packaging fragile items for shipping. Cells are fragile, much like eggs. They can be damaged by mechanical stress, immune system attack, or harsh chemical environments. The encapsulation capsule acts like bubble wrap, providing a protective barrier that shields the cell from external threats, while still allowing nutrients and oxygen to diffuse in and waste products to diffuse out. The microfluidic device is like an automated packaging machine that creates perfectly uniform protective packages around each cell, ensuring consistent protection for all. Let me address one more important question. What about permeability? How do we control what can pass through the capsule wall? Permeability of capsules depends on the molecular weight cutoff of the material. Small molecules like oxygen and glucose can diffuse through, while large proteins cannot. For hydrogels, the mesh size of the polymer network controls diffusion. Tighter cross-linking creates smaller pores and reduces permeability. Diffusion is governed by Fick's law, where flux J equals negative D times the concentration gradient. D is the diffusion coefficient. When designing encapsulation systems, we must carefully tune the permeability to allow nutrients in while keeping therapeutic proteins or cells contained. Let me summarize the key concepts we've covered in this lecture. Microfluidic encapsulation enables precise trapping of materials in protective barriers at the microscale with exceptional control and uniformity. Low Reynolds number laminar flow is essential for predictable, reproducible droplet formation in microfluidic devices. The physics is governed by interfacial tension, capillary number, and surface wettability, which determine droplet size and stability. Multiple device geometries exist, including T-junction, flow focusing, and coflow, each with advantages for specific applications. Applications span diverse fields, from single-cell genomics to targeted drug delivery to regenerative medicine and tissue engineering. The future holds tremendous promise for creating artificial cells, smart responsive drug delivery systems, and advanced diagnostic platforms. Thank you for joining me in this detailed scientific exploration of encapsulation in microfluidic devices. This field continues to advance rapidly and holds enormous potential for transforming medicine and biotechnology.